Hey everyone, I'm very excited today to introduce Dr. Marion Koopmans, who is a renowned veterinarian and virologist from the Netherlands and has a very impressive list of qualifications that I'm going to try to summarize. She's the director of the Department of Viroscience at Erasmus Medical Center in the Netherlands, the WHO Collaborating Center for Emerging Infectious Diseases, scientific director for Emerging Infectious Diseases of the Netherlands Center for One Health, and scientific director of the Pandemic and Disaster Preparedness Center in Rotterdam. Her research focuses on emerging infections with special emphasis on unraveling pathways of disease emergence and spread at the human-animal interface. And during the COVID-19 pandemic, she played a pivotal role as a member of the outbreak management team that advised the Dutch national government's response. She has a biography co-written with her son and has co-authored more than 700 articles that have been cited more than 40,000 times. We are so excited to have you here. Thanks so much for joining us. And please welcome.
Yes, thank you. See some familiar faces. Hi there. Yeah. So, okay, let me see. Yeah, presentation mode. This is good, right? Yeah, what I decided to do is to walk you through, two One Health-centered initiatives that we have been trying to establish here in Europe that really are trying to look at a more fundamental way of disease emergence prediction. I'm not saying we're there, but I just want to share the thinking and some of the work that is going on. So it's a broad topic, but just to hear also your views on this. And of course, the reasons why are very obvious. You are in the US in the center of one of the reasons why the weird change in avian influenza epidemiology and ecology when the virus entered the bovine population. We are still looking at Mpox spread both outside of the African region and inside of the African region with different, resulting from different spillover strains. And there's these other types of examples. And it's clear that this is not over and will not be over and is a consistent pattern that we see. But and of course, a key question is how do we know what to prepare for? And if you look at that and look at reviews of emerging disease outbreaks, but also look at the many different virus discovery studies, of course, we know many emerging infections are of animal origin, many are viral, and although many new viruses are being discovered, usually they belong to one of the families that we already know. And some of those families are notorious for spilling over and causing outbreaks. So really, if you look at it that way, zooming out a little bit, the list of candidates of virus families that might trigger epidemics seems finite. So we could potentially prepare for them by studying related viruses and developing model systems. And the key question is, if you then move closer into the ecology and trying to understand what is out there, then of course, questions are, can we predict? Can we predict which viruses can jump species? That's one. But also then once viruses do jump species, can we predict which ones will potentially also transmit among humans and how efficient that is? Very simple questions to ask, very challenging to really do properly, as we have all again seen during the COVID pandemic, but was obvious during the 2009 influenza pandemic. Now, to me, it's clear that if you have a pathogen that has to become a pandemic, certainly if it is moving as fast as we have seen with the COVID-19 pandemic, that's probably not something that is stoppable. But what is potentially stoppable is some of those more intermediary outbreaks, which is quite a few of the outbreaks that we see, that also serve as many examples and lessons to practice our ability to detect them early, and contain them, and maybe even learn to predict where the likelihood of these outbreaks is increasing. You all are familiar with the whys, and that is really that we are seeing lots of change in red in every aspect of the way globally in demographics. Political landscape has been on this slide, and is more to the point than ever. Climate, of course, land use change, and if you have change in any ecological system, you have to start thinking about what that changing balance and the change in interactions between humans, animals, and ecosystems potentially does to the risk of new diseases, or maybe in creating tipping points for known diseases. Now, what we are interested in here is really looking at this from focusing on what you could call pre-emergence. So if you look at many of the pandemic preparedness programs, there's a lot of attention to what do you do if once there is a pandemic, or I have a cat that is playing up here. Anyway, if there is a pandemic starting, or a large-scale outbreak starting, in terms of diagnostics, the genomics that really has moved to central stage, and all the data that you need for modeling and for public health decision making, vaccine development and what have you. But when thinking about being there at the earliest possible occasion, it really is important to start moving to the pre-emergence phase, and to try and understand dynamics of the different diseases as they are in animals, undulating around, and looking at trying to understand what drives a spillover, and can we use our understanding of the ecology to predict where the risks are, so that we can detect early. That's the sort of the central idea. So this is also the central idea in the VIO consortium, which is a European funded, Horizon 2020 funded program with partners across Europe, where we are trying to see, can we develop pre-epidemic One Health intelligence to be in the right place at the right time, to potentially pick up changes that might be associated with increased disease risk. So we work around that along 5 change scenarios. One is on vector-borne disease emergence, zoonotic wildlife-borne disease emergence, climate change scenario, one that we call silent epidemic, which is really a scenario that looks at spread through high density urban areas. And then the 5th one is the scenario that integrates what we've learned from the individual pathways. So what we're trying to do is see what we can learn if we start to move away from the classical, let's say, the public health surveillance data, that's this gray box here, but add in other types of information that you could call contextual data, that is information that could inform about drivers for disease emergence. So there's time, temperature data, demographic data, including movement data, here trade, travel, what we know about farming practices. So all sorts of factors that we know impact disease ecology, where there may be data available. It's not always the case, but where we're trying to explore how to use that data to help understand the ecology of diseases and the potential risks of spread. And we combine that also with catch-all tools like metagenomics and systems serology to try and bring that data together to learn. That's essentially what it is right now. I'm not saying we're there. So for each of the scenarios that I mentioned, what we are reviewing is what are the key drivers that are associated with this scenario, which data is available and in which structure, and what is the quality of that data, and how do we provide access to that data and the ability to search it, and can we or do we need, are there important gaps where we can bring in new additional types of data, for instance, through citizen scientists. And one of the things that we are developing is because the driver data sets are quite long, but can be used for multiple scenarios to study. So one of the things that we plan to end up with is an easy access tool to access all of the data types that are considered to be of sufficient quality to use and to integrate in analysis. Now, one of the emerging scenarios is exploring a specific climate change question, and it is focusing on Arctic, because that's an area of very rapid change. And of course, the Arctic is, depending on when and where you look, is a big part of the northern hemisphere, and is undergoing a lot of change. First of all, in terms of climate, it's one of the fastest warming parts of the world. There's plans for increased mining. We all are hearing about that these days a lot. Increased shipping, new transport routes, new agricultural land, tourism, and potentially new animal migration routes. With the big changes that are foreseeable here. So if the climate change continues as it is forecasted, there will be massive ecological change in this region, both natural but also resulting from our human activities. And there are already questions about issues associated with the thawing of permafrost, because it's known that permafrost and ice, so permafrost layers and ice, can contain microbes and viruses. There is evidence of metabolically active bacterial communities with infectious bacteriophages, so there is microbes that survive. There is, of course, the potential for highly stable pathogens that also are animal pathogens or human pathogens. Their concerns are, of course, because of the eradicated viruses, both for humans and for animals. Big questions are, what is the likelihood of retaining infectiousness during the process of release? Now, of course, permafrost, for instance, will not thaw just properly as you thaw a sample from a freezer. It will undergo multiple freeze-thaw cycles, so that will probably do away with a lot of pathogen infectiousness, but there are still questions about what can remain. Now, that debate has been kicked up a little bit when a study found that there are actually infectious live worms present in very old ice samples. So that really provided the proof of concept that there is the possibility for survival of even a complex organism like a worm here in a very ancient frozen material. And that frozen material can be very ancient. So there's now a record ice core that has gone down back to 1.2 million years ago. And then of course it's interesting to think, okay, with that, but certainly if you melt ice, what else could be in there? There's mention of some viruses that were found. And of course, people then have to sensationalize this a little bit with the zombie viruses. I think that's not very smart. But there is clearly the potential for some microbes and viruses that could be in there with retained infectiousness. So one of the things that we do is study ancient Arctic settlements that really give you a walk back through the human history of Greenland in this case. So this is with Greenland and Danish partners who they are archaeologists that have studied a couple of historic sites where Inuit populations lived that ended somewhere in history. And what these communities had was places where they dumped all their trash, including dead animals, waste, and potentially also dead humans, in the permafrost. So those middens, as they're called, are intact, are recognizable, and are becoming visible at the surface. And depending on where you are, you can have very recent waste sites and very ancient waste sites. So this is one of those layers where these archaeologists really can date them based on their knowledge. And as you can see, this is a layer that really is surface exposed, and this is permafrost that is starting to melt. And in those places, what you can identify is where farms are, of course, then where animals are near the farms, that's called infield. Their animals are further away, and then the earth above the middens. And these middens, as I said, are ancient, some of them. And what the group here does is just look at what does the microbial composition look like in these different places. Now here's an example of that work, which is really ongoing. This is bacterial community composition, and we're also still working on viral community composition. And what you can see, the colors reflect different middens, is that there are different communities present that also are different from the modern day microbial communities. Even so many years ago, and this is from permafrost material, not really very deep, so about 1 meter into the permafrost, collected into the permafrost. So the group is really trying to characterize these bacterial populations, including the possible resistance genes in there, just to understand what it is that is surfacing from these middens. And we are starting to look at the viral population. Now, there are other issues with this Arctic warming, in that there is really a very rapidly and expanding areas of grassland, which then really have an impact on the animal populations in there, and particularly the migratory bird populations. There's also effects on vector populations, but I won't go into that today. So one of the questions we were interested in is what could that retracting ice and increasing grassland do to migratory birds. And here, of course, we've seen a very abrupt and wild ecological shift when H5N1 viruses that historically, highly pathogenic H5N1 virus that historically have been circulating in mostly in Asia, really started to travel the world around 2020. So this is data on records, notifications of H5N1 highly pathogenic avian influenza in Europe from wild birds and from domestic birds, where prior to 2019 and 2020, we used to have occasional introductions, and they would be introductions of highly pathogenic H5N1 from Asia through migratory birds, and then also leading to poultry farm outbreaks. But the typical pattern was that would happen every now and then. The farms would be culled, the virus would be gone, problem gone. So that's been the pre-2020 situation. But that completely shifted in 2020 when these viruses, H5 and X viruses, really started to spread through wild birds, and this time across Europe, persisting in Europe, and then also skipping over to the US, to the Americas. And that's what you can show here. So a drastic increase in the numbers of observations of these viruses, drastic increase in the numbers of poultry farm outbreaks, and also, as you can see, no longer a pattern of interruption, but a permanent presence of these viruses in the wild. So big shift in the ecology. And that's where the Greenland site and also Iceland are interesting, because that's an area where you have birds. These are some common geese species that breed there, here in West Greenland. And these are birds that actually belong to the flyways, the bird flyways over the Americas. And these are birds that belong in the flyways over in the Eurasian flyway. But Greenland is a place where the two meet and where you can have crossovers. And that's, of course, that's also where we have done expeditions. This is one of the teams. This is their food for a month, meticulously packed. And this is the only project where I had kayaks on the budget. But this is where the team was camped out to go out and try and collect samples from geese. That's shown here. So there's geese, and that can only be done in a certain time of the year when they are changing their feathers, and they cannot fly as well. So they are easier to catch. And what we then do is catch them, measure all sorts of things, take swabs, take blood, take oropharyngeal swabs, and collect ectoparasites like ticks. And all of that material is taken back for testing. And one of the findings that we had there is that indeed it looks like both Iceland and Greenland have been hop-over sites for H5 influenza viruses. So in between expeditions, we saw the change in exposure to H5, also the presence of the virus, of the H5 virus, in the geese populations there where the two flyways actually meet. And based on that combined information, reconstruction is that indeed these places are where the H5, so it's sort of a highway for H5 virus dispersal. And the question, of course, is if this the suitability here for grassland and the birds that like that increases, what will that do? Will that lead to further dispersal of these viruses that also are present in Asia? So what comes with that is, with expanding viral habitats, is also expanding opportunity for mixing. So we've been historically, or not so long, so since the mid-1990s, we've been looking at Asia as a source for our H5 avian influenza viruses, and also as a potential pandemic threat. And in Asia, we've seen how these viruses have massively reassorted with resident wild bird and poultry viruses into an eclectic mixture of different reassortants, all with the H5 subtype. Now, the same is happening in Europe and in the US. Which makes it very difficult to really understand properties of the viruses that are being generated. So I think our H5 preparedness and trying to understand viral traits has really become tremendously more complex. Here is a movie of how, using the genomic information and looking at observations from different bird ringing sites, and combining that with locations, you can see the pattern of reassortants coming up. This is from Europe, so this is centered around Europe because it is a European consortium that studied this. But you see from the colors that different types of reassortants emerge, that we seed them back to Asia by now, but of course they also cross over to the Americas, where new reassortants arise. So quite a dynamic picture. Now, one of those viruses made it into cattle. That's you all know about that. One specific H5 sublineage, the B3.13, pretty wild, because before that, cattle were not considered to be very susceptible. Still unclear how that happened. Until recently, it was thought to be a single introduction, maybe some kind of freak incident. But of course, just about a month ago, a little more than that, we've seen another introduction of another sublineage, D.1, that and possibly 2 times now. So that raises the question, what exactly is going on? Why are these viruses introduced? Is this a viral trait that is different, or is there something in the process happening in cattle in the US that increases that risk? There's been testing across Europe, and so far no one has found any evidence of a similar situation in Europe. But of course, it is a concern what's going on there. So because of all those observations, so what we try to do is also link that then with, okay, now you see this, so what do you do with it? So we have another consortium in Europe that is now, there's all these new organizations and structures since the pandemic. This is funded by a sort of an emergency response type organization that has been developed. And what we have discussed with them is what you would want to do given the situation that we're looking at now with H5N1 in the US. So one of the things we have recommended is, let's start really looking at, if this virus would turn into a pandemic, or a variant of it would turn into a pandemic, what can we learn now possibly about baseline immunity that is present? And because that's, of course, what we've learned from the previous pandemic of flu in 2009, that maybe we had been staring a little bit too much at very specific correlate of protection, which was antibodies directed to the ahead of the hemagglutinin, the HI antibodies that sort of prevent docking of the virus. But in 2009, it became very clear that there's a very important second category of broadly reactive antibodies that targets the stalk, that does not completely prevent infection, but that does have a significant effect on flu severity. And that is, then the question is, do we have evidence of that also already present in the community? So this is work that's ongoing in just a healthcare worker cohort, people that have been part of studies during the pandemic, but that have agreed to continue to do so. So they are healthcare workers that are happy to be sampled every now and then. And we sample them just when they have a disease episode, but also on regular intervals as for monitoring. But then also when there's, for instance, a flu vaccination or a corona vaccination, or as we are currently doing an H5 vaccination. And they are sampled quite extensively, so we collect blood and cells and respiratory samples, including samples that allow testing looking for mucosal immunity. Now here is one of the data sets that we have generated, and this is looking at binding antibodies to the whole battery of flu virus variants. Here, these are all different H1 viruses from old to most recent. These are H2s, where of course you don't expect much of anything. These are all H3 viruses, influenza B, influenza H5 viruses, and here an H5 stalk. Not the top part of the hemagglutinin, but the stalk part. And here H7. And from this, you can already see that, hey, there's no binding to the head of the hemagglutinin, so no antibodies that you would see as protective, but there is definitely, and there's high levels of antibodies to the stalk of H5. And that's one element. So that's binding antibodies to the to H5. But then the second category is antibodies that bind to the second spike, the neuraminidase. And here you see that there's really very high levels of antibodies to those, to avian N1 proteins, and not a very distinct N6 protein. So there's clearly a level of antibodies present in the population, in this population, that in studies done previously and also in animal studies has been correlated with some level of protection from H5. So we think there may really be, that may be an explanation why the disease manifestations have been relatively mild. Of course, it's difficult to say with certainty. And right now we are doing this really in an age-structured cohort to try and see what we can find there. Then briefly, I don't know what time it is. I still have time. Yeah. So then one of the elements, of course, is looking at Climate sensitive diseases. We are seeing that increasingly coming up in Europe. You may have heard that we are by now every year have localized dengue virus outbreaks, even though we have very limited establishment. But looking at the frequency, for instance, in Southern Europe, in France, this is now every year. So it is, there is a concern that the current approach, which is really detection and containment, will at some point start to fail. And that is looking at, of course, this is a slide, an image from a review that was done by the University of Hawaii that did a literature review, including over 60,000 papers, asking the question, for what types of elements of consequences of climate change, by which mode of transmission can we see which possible consequences, and will they be increasing or decreasing? It's a very beautiful review, I think. And what you can see is that there's, through a mixture of routes, you can expect more vector-borne disease, that's obvious. Vector-borne, mostly viral, more waterborne, mostly bacterial, and so on. So a lot of potential change, but nothing that really is, it's not an easy picture that you can say. So in the region where we are trying to explore that as well. So what is our baseline, and what would it take for us to start seeing outbreaks? And we do that studying two flaviviruses. They are both mosquito-borne, not Aedes mosquitoes, but Culex mosquitoes, which is our common house mosquito here in Northern Europe. They are closely related. They both come from the African region. One is a virus. They both have the same birds that they seem to be infecting, although that's not entirely clear, and have started to circulate in Europe in a not so well in the past decades, with Usutu virus mostly affecting and killing birds, particularly blackbirds. It is a zoonotic virus. It is seen occasionally, for instance, in blood donors, but there is only a handful, I think 15 human cases that have been described, and these are mostly in people with immunity problems. And then there's Westnile, you know that very well, which of course has exploded in the Americas some years ago. We have that in Europe. We have repeated incursions, and it's quite a common cause of disease now in Southeast Europe. And our questions are, of course, again, if you think about these change scenarios, what can we expect for these viruses? Where could they move to? Can we learn to track that? Now, again, this is a very complex puzzle, because you need to understand the whole entire ecology if you want to, even try to predict where these viruses could go and where there is a risk of outbreaks. And these are some of the factors that, of course, you need to take into the picture. So it's complicated, and it's, that's throughout. We are really exploring to see what could potentially be leads for some real work into prediction. But again, here what we use is this driver-based framework, asking what are the key drivers, which data is available, what's the quality of that data, and what are critical knowledge gaps. And then you can make these complex graphs. This is what the APHA in the UK has done of the factors that you would like to take into account. And again, here we have then collected data both from viral genomic sequencing with data from birds, from dogs, from horses, from people, and from mosquitoes collected throughout Europe to try and see, can you reconstruct the movement of these viruses? And this is for West Nile lineage 2, where what we learned is, so we thought West Nile was constantly and zootic in this part of Europe. But what we learned is there's actually a region here and more in Central Europe that seeds these viruses continuously to the south and a different sublineage to the north of Europe. So yes, you do need control programs here. For instance, in Italy has many cases, but something seems to be happening here that could be fundamentally changing the dispersal of these viruses if we could get a handle on that. That's one of the observations. Then a question that also is looked at, and this is led by a group in Edinburgh, is trying to combine the genomic information with data sets about bird pathways, mammal species richness, land use, and what have you, and particularly here the farmland density seems to be very positively associated with the direction of spread. Potentially because that does attract some of the reservoir birds that are there. So that's what we are using now to target, for instance, the West Nile surveillance in our region. And that's shown here. So based on these findings, what we said, okay, let's do a deep dive in the ecology in the Netherlands. This is a big group of people that were happy because this was the first day out of lockdown after a long period of time. And what you're seeing here is a big field expedition collecting ticks, mosquitoes, sampling birds, sampling poultry, and sampling songbirds. And from that, so we've been doing that for a couple of years already. So from that, what we see is there, if you collect dead birds, there's a lot of Usutu virus there. We have never found West Nile there. In live birds, big numbers tested, there is Usutu, and we have found West Nile. And of course, both viruses have been found in mosquitoes, and particularly the West Nile came as a surprise, as was not expected, but clearly is there. So then, if you look at the phylogeny as well, this is for Usutu. We see two clades, one that is clearly enzootic. It's present all the years that we have been sampling. The other one more patchy, so that seems to be viruses that are periodically introduced. So two different patterns here, enzootic, and here introductions, that so sparks, as I call them. And here then is the same is for West Nile. So that was picked up in 2020 in different birds, in vectors, in mosquitoes, and it seems like that had not been introduced very long ago. Had not been observed in people, but once we then notified the public health system and neurologists were warned, it turned out there were several people hospitalized with encephalitis that indeed turned out to be West Nile cases. Since then, that has not expanded as far as we can tell. So we do have follow-up studies, including sentinel chickens and repeated sampling, from which we know that the virus is there, is lingering, and And the question is, what does it take for it to start causing outbreaks? There's a big difference between West Nile and Lesotho. Lesotho with explosive outbreaks, West Nile not. And we're trying to understand why that is, and if we can learn what it would take for West Nile to take off. And here's one of the examples, because in our country, which is a very low-lying country, what we're seeing is massive land use change, nature-based solutions, trying to prepare for sea level rise, for flooding, for severe weather events. So what you see here is projected wetlands that have been developed around the major cities, really to allow the water to go where it needs to go with the climate effects that we are expecting. And none of these programs so far think about the potential for vector-borne diseases. So right now we are starting joint programs and saying, okay, what does this actually do? So we are starting to, so we have a polder an experimental polder that we will start flooding this summer to really look at what effects that has on the mosquitoes and the bird populations, and whether there are problems with it and if it can be done differently. And it's really interesting because you will have engineers that manage these wetlands that say, oh, well, but then just tell us how fast we need to empty them again. It's really it is close to something that could potentially be actionable. So that's fun. So then a few words about the last arm, and that's the what we call the silent epidemic scenario, and that's our ability to pick up something that goes under the radar, because because there is a lot of mild or inapparent disease, and that's of course where we have seen the interesting applications of wastewater sampling. And what we already had in place is a global sewage surveillance project. So that's looking at the composition of wastewater in high density urban areas. This was not focused on any specific pathogen, but really what is the fingerprint, and can we recognize the fingerprint? What is the baseline, so that we can recognize deviations of the baseline. So this is samples collected across the globe, and in some places also longitudinal samples, and then just sequenced with metagenomics. And what you can see here is these are all individual virus families, from vertebrates, bacteriophages, viruses infecting archaea, plants, insects, and so on. Two methods of metagenomics. One is just really unbiased, whatever is in there. One is captured, which enriches for viruses that we are also interested in. So these are mostly the enteric viruses. And you can see that there's more of them here. So these are astros, calices, and so on. I just put this in because of Ben. So, for instance, this allows us to do a global snapshot of the diversity of noroviruses in all of these places. Here, looking at the broad genogroups, and here looking at the different genotypes, where you really see, you can get a one-page snapshot of what is out there circulating, and also where are there exceptions. So here were some unusual patterns picked up in Quito, and in this case here in Regina. G3 viruses in Melbourne. We think they may be of sheep origin. And here an example of, if you then dive in deeper and do phylogeny of the individual genotypes. Here's for one genotype where there was a branch that only contained sequences from African region. those were then cloned and used in binding assays, and they turned to have a very unique ligand binding pattern. So there may be really a genetic susceptibility question here. Just an interesting example of how you can even find infections that we didn't know about and learn from them. So that's just a flyover of the type of work we are trying to do. I do think we need to try to figure out to be better at early warning. That's what this is all about, because our response to outbreaks really is challenging. And we try to do that by gaining a deep understanding of the ecology. And the rest is, well, what I've just told you. Just to let you know that this is big endeavors with many people. The VIO consortium is in its final year. It's a real fun consortium of people that has been playing with this type of data. One Health Pact is in the Netherlands, and Durable is the new HERA-funded network that is there for pandemic preparedness.
Thank you so much. That was such an interesting talk. We're gonna open it up to questions. So I think it might be easiest if people just raise their cyber hand on Zoom, and I'll call on people to ask questions. While we're waiting, maybe I'll ask a question. And I'm not a virologist, so I apologize if this is a dumb question, but I was wondering for the wastewater surveillance, if that is something that can be used to identify novel emerging pathogens, or do you kind of have to know what you're looking for in order to quantify what's happening?
Well, a lot of it now is focused on what we know. And then, of course, if it's in the wastewater, it's already there, right? So it could be used to identify pathogens, novel pathogens that are circulating to a certain extent, because otherwise you don't pick it up in wastewater. A good example, I think, is what has happened in the US with H5. So quite rapidly, several groups have started to just include H5 in their wastewater testing, and they started to pick it up, also even in states that had not yet reported H5. So I think that's a very powerful example of how this really can help, figuring out what's going on, even if you don't know what there were discussions. What is the source of the H5 in the wastewater? Is that milk being dumped, or is it actually really H5 in people? There's no evidence of that. So there's uncertainty of how exactly to interpret, but it does give you a powerful signal that says, hey, there's something going under the radar. We need to figure out what is happening here. Now, that's the known viruses. But the reason why we're doing this fingerprint is to see, because there's a lot that you can then annotate, but there's also a lot that you cannot. And so that's the dark matter. But where we're now trying to figure out if we can use that, not knowing what it is, we think it's viral because of the way the samples have been prepared. But can you find clusters? So could you potentially find clusters, not knowing what it is, but seeing, hey, there's clusters developing. So that's what we're working on now. That's really very much exploratory, but in theory, it should be possible to identify clusters of something, sequence, in different types of different places, different samples, linked animals, people, without knowing what it is. And another set of studies that we're doing is enriching the materials by trying to do pull down with serum from different animals or from humans, so that you pull down stuff to which there is an immune response. So it's all trying to explore how you can work with these new tools to help prepare for you know, to help be faster in disease detection.
That's super interesting. Thank you. I think I saw Ben's hand and then Katya.
Thanks, Maria. Thanks, Marion, for coming to Emory. It's good to see you. Next time, physically come.
Would have loved to be there.
Yeah.
Would have loved to be there.
Yeah, we'll get you here. I want to kind of pick up on where you were kind of going with your reflections of the last question. You talked about how we have to embrace all this, how we have to embrace complexity in thinking about emerging pathogens, and you kind of gave this terrific overview of like all sorts of problems and data sources, serology, like environmental wastewater and human surveillance. Like, if we could be better at integrating or linking, like, one linkage of two kind of types of data streams, where do you think we would like make the most impact if you could link, have like really solid surveillance linked from it to narrow down some of this complexity?
I think it is, there's not one, I love the wastewater stuff, particularly if you can also think about doing it in, animal slaughter facilities, in transport hubs, doing it in Rwanda, DRC along the highways to see can we track what's going on there. So that one combined with so wastewater is of course embraced now by many, but I think working with the wastewater data is also a little bit stifled by discussions about validation and about individual targets and detection limits. I think the best way forward is to start embracing that this is a novel type of data that does not really fit the mold of how we have worked with laboratory data in surveillance. But just say, let's consider it experimental data that can give signals like you do horizon scanning, where once you see something, you would really want to look at a second source to confirm, but not trying to fully validate it. There's no way you can validate metagenomics to the details level that you do with regular laboratory tests in surveillance. So that's one. So I think the metagenomics wastewater as a novel type of information, bringing that into the routine types of data collections that you have in all sorts of investigations, I think that would be a very good step. And the second one is that I didn't mention it now, but we are, for instance, in the mosquito work, working a lot with citizen scientists. So citizen science data is also a little bit, it's not fully reproducible. People love it for a while, then they're gone. But it has given us some of the best information, for instance, on the mosquito work. So those are two that I really think are important. The citizen science also because it has a potential educational effect. So it's a two-way, a dual purpose approach. Yeah.
Katya, go ahead and you'll be our last question.
I have a couple of much more, I guess, specific questions. So one was going back to your HA and NA titers. I'm wondering whether you looked at the NA titers with the human serum and kind of the avian NAs. I'm just wondering, have you done the same for kind of just the human subtype NAs, and what was that magnitude difference? Were the avian NAs of N1s very similar to kind of the human
Yeah.
ones?
Very similar.
Yeah.
Slightly lower, but very similar.
Okay, but on the same order of magnitude.
Yeah. Yeah.
Another question I had was actually more on the permafrost, the PCA for the permafrost data, which I found really fascinating. And I'm wondering, I don't know if you have time to kind of, show that plot again, but it seemed like the colors were the different locations within Greenland, and the markers were the different kind of effectively time periods, like the midden versus the different areas within kind of location. And just by squinting, I couldn't really tell whether the markers actually within the same color really kind of overlapped. So where do you, within a certain region like West Nuuk, do you actually see systematic differences kind of between the midden and samples from other kind of I guess, the
more recent
exactly.
Yes. So the observations so far seem to say that the soil on top of the middens is different.
Yeah.
The same animals walk there, the same thing, but the soil is different. The soil composition has remained different. So that's, of course, quite remarkable.
Yes. Great, thank you.
Yeah.
Fortunately, we are at time, but Marion, thanks again so much for joining and for giving the talk. That was fascinating. And thanks everyone for your questions.